Hello, everyone, and welcome to today's live webinar broadcast, hosted by Samsara Sciences and presented by Dr. Sharon Presnell, President of Samsara. My name is Denise Weiser. I'm the Technical Sales Manager here at Samsara. Before we get started today, I'd like to remind everyone in the audience that we are broadcasting live, and following the presentation, there will be a Q&A session offering you a chance to pose questions to our panelists. We encourage you to submit your questions at any time throughout the presentation by typing them into the questions pane of the control panel. We will collect the questions and address as many as we have time for following the presentation. I'd now like to introduce Dr. Sharon Presnell. Sharon holds a PhD in experimental pathology from the Medical College of Virginia and completed her postdoctoral training at the University of North Carolina at Chapel Hill. She has 20+ years experience working with primary cells related in vitro and in vivo models and has worked across multiple organ systems, including liver, kidney, and pancreas. She has been responsible for research and new product development at Becton Dickinson, Tangian, and Organovo, and started Samsara Sciences in 2014 in response to unmet needs in the liver cell supply market. Welcome, Sharon.
Thanks, Denise, and thanks to everyone who joined us today. Hopefully this will be an interesting topic for you and you'll have lots of questions for us at the end. We're going to cover today the isolation, characterization, and use of the donor-matched primary human liver cells that we work with at Samsara from both healthy and NAFLD NASH donors. And so with that, let's get started. So we're going to go through a brief introduction. We're going to talk a little bit about our isolation methods and the cells that we produce, how we characterize those typically. We're going to talk about the non-parenchymal cell's role in liver function and pathogenesis, and we're going to do some comparison of non-diseased and fatty liver disease NASH tissues, and that's going to include some immunochemistry and in situ hybridization work that we've done here. Also a pilot study with RNA-seq profiling and results from that. And then we're going to share with you a use case where we've taken disease origin non-parenchymal cells and worked with our parent company, Organovo, to build 3D printed human liver tissues with these disease origin cells and how that turned out. And then we're going to introduce to you a new service that we're providing where we're taking these donor-matched cell sets and providing RNA-seq data packages of those to customers who are interested. So the background here for folks who may not be as familiar, the role of non-alcoholic fatty liver disease in the population at large has been growing for some time, largely secondary to obesity and type 2 diabetes and other kind of lifestyle-induced epidemics within the developed world. And so if you think about the stages of progression of this disease, the liver goes from being a pretty normal liver, like you see here on the left, to being fatty and lipid accumulation within the hepatocytes. And then it moves on to NASH, where you begin with that bright blue staining that you see in the histology there to see some fibrosis. But at this stage, the liver is what is referred to as compensating, and is able to still function, at least to some degree, even though that fibrosis is present. But over time, that will continue to progress. If there are no lifestyle changes and no interventions, that will continue to progress to decompensated cirrhosis, where the liver really can no longer repair itself. So today, 30 to 40% of the adults in the United States, so 1 out of every 3+ of us sitting in the room, are on the NAFLD spectrum, and 20% of these are going to progress to NASH over time, and it's predicted to become the leading cause of end-stage liver disease and need for transplant in the coming decades. It's important to recognize that the initiation and progression of NAFLD are driven both by the genetic background of the individual and the environment. And so this is a really simple diagram that we found in a paper and are sharing here. But the idea is that if you have genetically susceptible individuals, and there are a couple of genes now like PNPLA3 and TM6SF2 that have been associated specifically with a higher risk or susceptibility for NAFLD and NASH. If those individuals are in a favorable environment, meaning they have a healthy diet and exercise and have some lifestyle management, they may have, even though they have this susceptibility, they may have either no disease or delayed onset of the disease. Whereas if you put those same individuals in a risk environment, so fatty diet and poor health in general, they have a greater risk of developing that disease. And the flip of that would be true for people who are not genetically susceptible. They may eat McDonald's every day and never really have a problem. But it's important to recognize that there are both kind of lifestyle, environmental influences to these diseases, as well as the genetic background of the individual, and that they're also very multifactorial. There's not one button that's getting pushed essentially to cause the disease phenotype to appear and progress. So on this slide, the purpose here is to really get into the different cell types of the liver and the role that they play in initiation and progression of disease. So in the beginning, the hepatocyte is accumulating lipid, accumulating fat, and this damages the cells over time. And there are multiple hits. So this could be an insulin-resistant individual. It could be an obese individual. They could have type 2 diabetes, hyperlipidemia. And then there can also be just poor diet where you're getting then endotoxin from the gut-liver axis popping into the liver on a regular basis. And so this causes the release of cytokines by the hepatocyte, also damage associated molecular patterns. And this is a profound irritant to the immune cells that are in the liver. And so you can get activation of the Kupffer cells that are in the liver in addition to recruitment of additional immune cells into the liver. And this inflammation then comes back and influences further hepatocyte damage, also activates the stellate cells, which go from being these nice quiescent vitamin A containing cells to more like an activated myofibroblast secreting ECM. And so all of these things acting in concert are what drive the progression of the disease. And so it's easy to think about when we try to put cells in a culture dish and study what is happening or how a drug might influence any of these pathways, that you might want to ask those questions with more than one cell type present, because there's such a multitude of factors affecting the progression of the disease. And so before we get into the individual cell isolations, just a little bit on liver microanatomy for folks who may not be familiar. So the liver here on the, let me just grab my laser pointer. This is the whole liver here, which is divided into lobules. And if you were to look at the liver histologically at this spot here or this spot here, the truth is you wouldn't be able to tell which spot you were in. It's pretty homogeneous in there. And the blood is going to come into the liver through the hepatic artery and also the portal vein, and that's going to come in through these portal triad areas and flow this way toward the central vein. So the blood flow is going to come in here and go there. And so these are the lobules. And you can also think of it like an acinus, where these three central vein areas here are going to be draining from this portal triad area here. So there's lots of ways of geometrically thinking about it. But we understand pretty clearly that the job of the hepatocytes here is pretty different than the job of the hepatocytes here. These are very active in protein synthesis and that type of thing, and you see more P450 drug metabolizing activity in these down here. And so if we go through, this is now an even higher magnification, if you will, of the lobule. And so these are the hepatocytes here, the gray cells lining the sinusoid, and you'll find the Kupffer cells sitting in the sinusoid where they're gobbling up stuff that you really don't want in your body. And then the stellate cells hang out here adjacent to the hepatocytes and the liver endothelial cells, which are lining the sinusoid. And these endothelial cells are pretty specialized. They're not going to be like a garden variety endothelial cells. They're very fenestrated and leaky so that they can let things in to the hepatocytes. And then the waste forms and flows in this direction into the bile duct here. And so when we're doing our cell isolations, we're after the hepatocytes, the stellate cells, the endothelial cells, and the Kupffer cells. So when we first get the liver, we do hook it up to some pumps and enzymes and digest this enzymatically to give us this big bucket of soup, which contains all the cell types. And this mixed suspension then is separated from the pellet. So we do some very basic centrifugation, and from the pellet, we get the hepatocytes. Those go straight from being isolated into the freezer. They don't experience any time in culture. And then we take the supernatant, which contains the non-parenchymal cells, and we isolate the Kupffer cells from that supernatant through a positive selection method. These also do not get cultured. They go straight from being isolated into the freezer. Then the stellate cells are isolated through density gradient centrifugation, and the endothelial cells through elutriating centrifugation, and both of these cell types go through a passage on in tissue culture before they are put away, and that's part of their purification process as well, to use media that selectively grow those cells and eliminate things that may have hung on during the process, if you will. So I'm going to go through some of the characteristics and things that we do to assess each of the cell types that we isolate, focusing on the non-parenchymal cells. And so here we're going to talk about the Kupffer cells. So when we finish the isolation, the cell population is going to be comprised of CD11b positive cells, a proportion of which will also express CD68, and some which express CD163, and there's not going to be a complete overlap in those populations. They are competent for LPS-induced cytokine release, and this is just a neat picture that I like that the team provided. So these are control cultures. This is the product that we provide, so after it comes out of the freezer and you've thawed and plated your cells. And so these are cells that have not experienced LPS, and these are cells that have, and you see the very characteristic elongation and kind of activation of the Kupffer cells in response to the LPS. And then we also know if we take these cultures and expose them to beads that have fluorescent particles on them, that the cells will phagocytose the beads. And this is just a really nice high magnification picture of this very large cell that has gobbled up several of these beads. And then we standardly do look at the Kupffer cells with an LPS response at the cytokine level. So we run the MSD 10-spot on these plus or minus LPS, and we look at 24 hours and 72 hours and calculate the fold change in cytokines, and also just really which ones are up. And we very frequently see interferon gamma, IL-1 beta, IL-6, IL-8, and TNF-alpha. Those are almost always the big players, and sometimes depending on the disease state of the individual, you may see some different patterns in that, but that is pretty standardly run to characterize the cells that are made. We also do for the stellate cells some very basic immunochemistry, just looking at markers, and we're going to talk a little bit later about some additional markers for human stellate cells that may be more telling of their activation state, because a lot of the markers that have been described in the mouse don't translate particularly well to the human, which is not surprising. But we do stain these for alpha SMA, and we also look at things like GFAP and Desmond, which will be positive in the population, but don't always correlate very well with activation state. And that's one of the differences in the human and mouse. And then we also look for fibroblast by TE7, and we do see occasional positive cells or weakly positive cells in that as well. On the endothelial cells, we do, and this is an advanced passage. So here, these cells have been grown out 3 passages, but we do this multiple times as the cells are being expanded and after they're isolated. We're looking at the cell surface marker profile of these cells, and we see very typically CD31, CD146, which will go down some over passaging because it does get internalized, CD45 negative, CD299 subpopulation there, and then there's also very heavily positive populations for Live1 and von Willebrand factor. And then the image here is just showing you what the morphology of these cells looks like again at passage 3. And we've done some experiments with these, looking not only by flow cytometry, but looking at immunochemically. These particular cells were grown and put under conditions of laminar flow. And one of the things that we saw is that the cells would line up in response to that flow, and that's what you would expect to see. We do see the CD31 positivity. We also see some CD32B and Live1 staining in the cultures as well. kind of validating that phenotype that they have. And then this is some really beautiful data that was provided to us by Andrea Rothmeyer at Organovo. So she's looked at these cells and compared them to a very kind of garden variety endothelial cell that is used for a lot of things, the HUVEC. And so this is a tube formation assay in Matrigel. And so what you see here on the top, these are just replicates of the same assay run at the same time. And so this is very typical tube formation by HUVEC in Matrigel assay. And then what you see with liver endothelial cells is that, and you would maybe not expect them to be exactly like a HUVEC, they make these beautiful astral structures, and they do form tubes and connect, but they do it in a very different way than a typical systemic endothelial cell. And then if you take something like a stellate cell, this is good proof that we isolate what we think we isolate. But if you take a stellate cell and put it in an assay like this, it does absolutely nothing. It just becomes kind of a clump and sits there. And so this is a really nice functional type assay to say these cells have the markers that they have, but they can also do something that endothelial cells are supposed to do, which is form these vascular tube-like structures. And so I'm going to move on now and talk a little bit about the characteristics at the tissue level of these diseased and non-diseased liver tissues that we work with. And we do take these formalin-fixed paraffin-embedded tissues and make tissue arrays from those so that we can look at things in kind of high density on our characterization runs. And so if you take something like, and this is a very low power image on purpose because we just want you to be able to appreciate, if you look at these are the healthy livers down here, and as you go across the slide, they're increasing their NAFLD activity score, which is the score that the pathologist that scores these tissues assigns based on the same clinical type. If a clinical biopsy were to come into the clinic, she scores these the same way that a clinical biopsy would be scored. And so as you go from left to right, the NAS score is going up. And so you can even see this is collagen in red and smooth muscle actin in green. And just looking at this very low resolution, you can see the differences that happen in this tissue over time. And a lot of that is being driven by the stellate cell activation that happens in the course of NASH progression. And you can appreciate that here at a higher level. So this is a non-disease comparator from the tissue on the leftmost side of that array. And then here is, again, the SMA is in red, or the collagen is in red and the SMA is in green. And you see collagen deposition, but you can also appreciate the ones that look kind of orange or yellow. These are the ones that the cell is SMA positive and also making the collagen 1 in these very fibrotic areas of the tissue. And then just blowing that up again for you to see the morphology of these cells and what an extensive network the collagen is forming around these hepatocytes that then really get walled off and unable to communicate with each other. And that's a big part of the progression of the disease. And you do still see some SMA alone and some collagen alone, but there's a lot of association of the collagen areas with the SMA positive cells. And so we've been very interested also in using these tissue arrays to look by in situ hybridization for some genes that we care about and want to be able to interpret in the context of, let's say we've seen a gross level, we've just looked at the tissue or by gene expression, and we've seen a gross change in something like reelin or smooth muscle actin. We like to be able to go into the tissue and from a spatial context be able to understand which cells and which places in the tissue are expressing those. And so if you look at the normal liver, the non-NASH liver, all of these little teal cells that you see here, these are reelin positive stellate cells lining the sinusoid, and they're spaced appropriately and in the appropriate numbers. And then here we really just see the one little ring of SMA positive cells. So this is the smooth muscle actin here lining the hepatic artery and the portal triad. And in contrast to that, if you go now to the NASH tissue, there's this area of fibrosis here, and you see lots of SMA positive cells. There's also very distinct changes in the patterning of the renin positive cells within the lobule. The lobule itself is disorganized. And so generally you might find more renin positive cells in a NASH tissue than you would find in a non-NASH tissue. But you also see this phenomenon, and I'm going to blow this up again. Here's a different case, just a different spot showing the same pattern. But now if we blow this up even higher, what you begin to see is cells that are double positive for both the renin and the smooth muscle actin. So the renin is the teal color, the smooth muscle actin is the red color, and they're almost always in close proximity to these lipid droplets. And so this is a really nice indication that at some point these renin positive cells begin gaining this SMA expression, and one can imagine that over time they simply become SMA positive myofibroblasts, which may or may not express the renin anymore. If we look at the immune cells in the tissue by CD68, so here we're looking at the CD68 positive cells and the non-NASH on the left here, and the green again is smooth muscle actin, just to give you some context, and then we're looking at them here in the NASH liver. We see a lot more of the CD68 positive cells and also can see them, we look at the higher power here, associated in these bands of fibrosis. And so this is very congruent with the literature in this space, and is an area we'll continue to explore with other immune cell markers. So we were intrigued enough with the differences in the healthy and the NASH livers to want to run a very small pilot study of RNA-seq, just to say, if we were to look at the whole tissue level, would we be able to see differences in gene expression that would potentially correlate with a non-parenchymal cell component versus necessarily a hepatocyte. And so we did a very small pilot study, 5 native healthy tissues and 5 native diseased tissues with a NASH score of 3 or greater. And this was run by a really great company called Gubra. They do a great job with the RNA-seq and the analysis. And at the end of the analysis, we found over 700 differentially expressed genes at a significance level of 0.05, of less than 0.05, that were different between the healthy and the diseased tissues. And principal components analysis is shown here. And even though it's a very small sample size, and we have this outlier down here on the disease side, you still get some separation of the normal and the diseased on the principal components analysis. And so this is directionally encouraging. We definitely want to look for more data, and we're going to talk about that subsequent. But we did take now this set of genes that are definitely interesting in the progression of NASH, things that are known. So smooth muscle actin, the collagens, MMPs, pay attention to, and then LOXL2 and TGF-beta, which are going to be stellate cell specific or stellate cell associated for activation, and then things like the TIMPs. And we looked at these in this data to see, could we see differences even at that whole tissue level in these key things that ought to be there as the disease progresses? And the answer was yes, we actually see them. And what makes this interesting, so you're analyzing the gene expression from the whole tissue, and the hepatocytes are 60 to 70% of that tissue. The non-parenchymal cells are 30 to 40%. And then if you break that down further, the sinusoidal endothelial cells are going to be about 50% of that 30 to 40%. The Kupffer cells are going to be about 20%. The lymphocytes are going to be about 25%. The biliary cells about 5%. And then the stellate cells are 1% of the 30 to 40%. And we're seeing things in this whole tissue analysis that would be suggestive of stellate cells contributing to that signal. And so what we are interested in down the road, which we're going to talk about in a few slides forward, what we're really interested in is understanding what kind of things might we see if we look at the whole tissue, but we also break that tissue up into its individual cells and RNA-seq profile the individual cells associated with that tissue. And what we hypothesize here is that the differences that are contributed on a cell type specific basis will become much more apparent and mineable in that data set. So one of the questions that we had very early on is if we take something like a stellate cell and we isolate that from a disease origin tissue versus a healthy origin tissue, is it possible for those cells to retain their disease features in vitro in the culture dish? And so this is a very simple kind of morphologic experiment, but we took stellate cells from a healthy donor and stellate cells from a NAFLD donor. And we do routinely see morphologic differences in these two origins. And so these are the cells from the healthy donor, and these are the cells from the disease donor, and they're much thinner, less stellate, if you will, and longer, more stretched out. So they have what I would think of as a more activated phenotype. And these are differences now that are maintained a couple of passages out. This is not the initial culture. And so if we look at things like Desmond and GFAP and SMA, the healthy donor cells have lots of the Desmond and GFAP, but a little bit of SMA. But then if you look at the disease origin cells, they don't have much of those, what have been considered classic stellate markers in the mouse, but they do have lots of SMA. And so we see some differences despite isolation methods being very similar. If we take now those cells and look at them gene expression patterns, these are the healthy origin cells, and the ones on the right are the disease origins. It's two different lots of disease and two different lots of healthy. If you look at TGF-beta, they're already making their own by a great amount here compared to the healthy origin. They're also making a lot of smooth muscle actin, and one of the two lots is making a lot of collagen as well. And so instead of thinking about taking this cell and hitting it with TGF-beta to activate it, you could imagine that this stellate cell is making enough of its own, and it's on its own road. It's making its own food essentially. And in more of an activated state. So we believe that it's possible for these cells to actually retain some of these phenotypic traits and functional traits after isolation. And so we're going to do just real quick a use case. So this is a really nice set of experiments done by our parent company, Organovo. And what they did was to take the process that they typically do. So they're taking primary human cells, putting those into what's called a bioink. It goes onto this printing platform, this bioprinter, 3D printer, and makes these little liver tissues in a 24-well plate. I'll give you a look at what those look like. So the input cells, and in this particular case, Kupffer cells were used. So Kupffer cells, primary hepatocytes, primary stellate cells, and endothelial cells. And then this is what the tissue looks like. It sits in a little transwell cup. And then if you histologically look at that tissue, it has some compartments in it. So there are compartments that are enriched for non-parenchymal cells and compartments that are enriched for hepatocytes. And this gives you the ability to keep the tissue around a little longer. So the durability of it is pretty good, and it maintains some functions. But these concentrations of non-parenchymal cells also enable you to see some pretty cool stuff like fibrosis easily in the tissue. And so this is, each of these little tissues is about 1 million cells. They're all touching each other, as you can see. So it's like not like you would see cells on a scaffold, for example, and it's maintained in this kind of standard system. And so there's a lot of biomass there. You can do a lot of experiments with each little tissue. And so in this case, what they did, instead of taking the Kupffer cells, the ECs, and the stellate cells from a normal donor, we use the same lot of hepatocytes. And so here's the tissue from which the disease origin cells came from. The Kupffer cells, LECs, and stellate cells were all isolated from this disease donor. They were combined with the same healthy hepatocyte donor that went into making this tissue. So this is the standard tissue made with healthy hepatocytes and healthy origin non-parenchymal cells. This is the same hepatocyte lot, but made with disease origin non-parenchymal cells from this donor right here. And so with no further stimulus, so no TGF-beta was added or nothing was added to this, tissues are just printed and maintained, you see this really florid fibrosis. And so what you're looking at, this is a low power image of the tissue. And what you're seeing down here at the bottom, all this blue is fibrosis being deposited, most likely from those activated stellate cells. This is a higher power image of this. And then if we go up here and look, the red is smooth muscle actin, which is very rich in this area where the fibrosis is. This area out here is mostly hepatocytes, because as we talked about before, this is kind of a compartmentalized tissue. And then if you look at the healthy donor, you do see a little bit of SMA, but in contrast, you also see a lot of desmin, and you don't see that fibrosis in this tissue. And so what this really pinpoints is that with only just using those disease origin cells, you're able to push a disease-like phenotype with respect to fibrogenesis in the tissue. And so this can be a really interesting way. You could even imagine mixing and matching the different cell types as to origin, to kind of figure out mechanistically which cells might be driving a particular process. So just recapping our current state of knowledge and kind of what we've covered. So the native, non-diseased, and NASH tissues really do vary widely with respect to their architecture, the histologic features that they have, and the gene expression patterns that they have. And many of those can be tied to individual cell types like the stellates, the Kupfers, and the endothelial cells, and they display very clear differences in the healthy versus disease state. The isolation of hepatocytes and the individual NPCs is feasible even from these NASH livers. And so this gives us the ability to create what we call donor-matched sets. So the feasibility that you can get the whole tissue plus the hepatocytes, the Kupffer cells, the LECs, and the stellates, all from the same genetic background. What this does, kind of two things. You're almost guaranteed biocompatibility because these cells have been living together forever, how old the patient is. And so they're likely to be compatible with each other. But you're also getting, if you have a donor, for example, that has advanced NASH and they are type 2 diabetic and they might have that PNPLA3 gene, what you're getting then is all your non-parenchymal cells of that same background, because maybe it would be naive to think that only the hepatocyte would be driving the disease. And so it gives the opportunity to do some, potentially really informative chimeric combination type studies, or, really begin to look at things at a deeper cell-specific level. And then we also are learning that these disease-associated features are retained in the disease origin non-parenchymal cells, at least to some degree, after the isolation and culture. And we can begin to leverage that in different modes. So I want to introduce now a new service that we're going to be providing. So we are going to be undertaking transcriptomic analysis of these donor-matched sets from NAFLD, NASH samples, as well as healthy samples to go with it. So the livers come in, and we always take snap frozen samples from the original tissue. And these also go to formalin and paraffin embedding, and a pathologist reads each of these. So we have a pretty extensive donor history report that goes with the liver in addition to this donor data and all the data from the snap frozen tissue and each of these different cell types that are isolated. And so what this affords us the ability to do is now take this whole tissue, replicates of the whole tissue, and replicates of each of the isolated cell types, and conduct RNA-seq profiling from that and provide that data. And when you elect to do something like this, you're going to be able to get the data at a much lesser resource requirement than if you had to purchase the samples and go and do it all on your own. And there has been enough interest in something like that for us to put it forward. And so there's two ways to access the data. We're doing two different things. So we've got 8 historic donor-matched sets that we are running through the RNA-seq profiles. 3 are non-diseased and 5 are diseased. And specifics are down here in this table. And each of these are going to include the whole tissue, hepatocytes that are uncultured but cryopreserved, Kupffer cells that are uncultured but cryopreserved, hepatic stellate cells at the end of their initial passage and cryopreserved, and liver endothelial cells that are at the end of their initial passage and cryopreserved. And so that data set is going to be made available in the very short term. And so I'll give you some contact information on the next slide where you can reach out to obtain if you're interested in that. We're also going to initiate a prospective donor match profile. So we're processing a couple of non-disease livers per quarter, and probably around 2 to 3 or 4 NAFLD NASH livers per quarter. And so here we have the ability to do something a little bit unique. So in addition to the samples that we talked about in the historic, so the whole tissue, and then each of the kind of products we provide today, which are these last 4 bullets down here. We're also going to be working with hepatocytes before they're ever cryopreserved. So the very initial isolate, the Kupffer cells before they're cryopreserved, the stellate cells before they're cultured or cryopreserved. So they will not have been on tissue culture plastic when they go for the RNA-seq. The liver endothelial cells before they're cultured or cryopreserved, same thing. They will not have been on plastic before they go for the RNA-seq. These will not be available as cell samples. They'll be available as data. And then these will be available as cell samples as well as data. And individuals who sign up for the data profiles will have priority access to these live cell samples as well. And so the package is going to include the full donor history report. This is all the pathology report and the donor data. And you'll also get all the isolated cell characterization data. So all the LPS response to the Kupffer cells, for example, the flow cytometry of the ECs, the tissue RNA-seq profile from the whole tissue, as well as the matched isolated RNA-seq profiles from the cells. And everything will be run in triplicate. We are using the Illumina HiSeq platform with ribosomal RNA depletion, not mRNA enrichment, and we'll be running everything on triplicate with an anchor sample included in every lane and then in all runs. The data will be delivered by a secure USB drive. And then, as I mentioned on the previous slide, when you access the program on the prospective side of things, you'll have priority access to the associated live cell samples to run in experiments with live cells. And we'll also work with folks on a custom basis if there are things that you would like that we're not standardly running. Because we have these unique samples, we're very willing to work with you and work what you need into the picture. If you're interested in any of these things, please contact sales@samsarasciences.com and Denise will take care of you. And with that, I'd just like to thank my colleagues on the Samsara team for all the hard work and for making it possible to talk to you guys today about these things. And we would love all your science questions.
Well, thank you, Sharon. That was very interesting indeed. We have a few questions from the audience, so we would like to begin the Q&A segment. But just to remind you, you can still submit questions through the question pane in your attendee control panel. So let's see. We have, does your cell isolation protocol change from healthy non-fibrotic versus NASH disease livers that are fibrotic? And if so, what are those changes?
So the short answer to that is yes, there's some tweaks that we have to make because it's, you can imagine that even just the digestion of the tissue is going to be a little bit different when there's so much fibrous matter there. In terms of the characteristics that we use to segment the cells from each other, so the positive selection or the fact that we might use a density gradient, that won't change. But sometimes you can imagine like the vitamin A storage in a stellate cell, for example. So that's going to change its buoyant density. And so the specific density that the stellate cells might appear in a gradient or an elucidator, that might be a little bit different. And there's no hard and fast concrete. I think we've got a team here who has a lot of tacit knowledge, and so they do a lot of optimization based on the characteristics of the specific liver. But yes, it would be fair to say that things have to be slightly different for a disease origin versus a healthy.
Thank you. Next question. How do you isolate LS, the sinusoidal endothelial cells, separately from the stellate cells in the same non-parenchymal fraction?
So the elucidator allows separation of those populations, and so you can we can separate them, the ECs and the stellate cells, through elucidating centrifugation. We don't always do it that way, but the endothelial cells appear in a different fraction during the elucidation in the stellate cells.
Thank you. Great. Next question. How do you get access to diseased donor tissue?
Well, we take advantage of that 30 to 40% statistic that we talked about at the very beginning of the presentation. So because such a large portion of the donor population has NAFLD NASH, and they're on the spectrum, we take those donors in because a lot of times the disease is advanced to the point where they are not really good candidates for transplant anymore. And rather than have that organ go to waste, we're going to take it in and isolate cells from that. But they come through the same channels. So they have the same level of consent, and they come through the same channels through the organ procurement organizations.
Great. Can you give us an idea of how many cases you've processed to date?
So I think we're up to liver 80 something in a couple of years of operation. And so we are growing. We're in kind of a small company getting a little bit bigger. And so our number of tissues may increase over time, but I would say, it's fair to say that we're going to be processing between 25 and 30 cases a year.
What applications are your cells being used for?
So we don't always have insight into everything that customers are doing with the cells. There's a few things that we do understand. Certainly, the birth of our RNA-seq profile service is coming from the realization that that's what a lot of people do with the cells when we buy them, and we're trying to create a more economic way for them to get at that data by offering the service. And so we know that's a big part of the effort. And we know some things, like even internally, being able to use the stellate cells and look for something like a TGF-beta response. People are incorporating multiple cell types together in some of the new generation models. They're really exciting. And those are the things we know about today.
Okay.
Yeah.
Thank you. With some of your data here that you're collecting, have you noticed any patterns or anything so far, even in your short sample size, that would be of note?
So we do track the characterization data that we have. We do track that back. So everything from the donor demographics and the donor data all the way through to the cell characteristics of the isolated cells. We have begun to build a database of that and look for correlations in that data. And we are going to be presenting some of the initial data from that work at AASLD this fall. We're really excited about that, but we have had some learnings on that front. So things like BMI, things like donor age can influence in a unexpected way cell doubling time, for example. And so we're learning some of those correlations, and it's a big interest that we have going forward to begin to take the RNA-seq data, the donor data, the cell characterization data, and be able to create the type of database that enables us and our customers to mine that for things that all help us kind of get better at our jobs.
Interesting. How much of a concern is there for product consistency in the face of vast genetic differences between liver donors over time that will make even same disease state or non-disease packages give different results?
Yeah, I think this is really one of the great challenges and opportunities as we see it, because I think I have, speaking for myself personally, I think I have given up on the notion that you can take 5 human beings and make them homogeneous. I don't think that's going to happen. And so we've begun to think about donor heterogeneity as a reality. And how do we use that and leverage that to the best that we are able? And that's a big part of the database building that I was just talking about before. But we hold no illusions. I really believe what we'll be looking for is to, over time, continue to grow this data set. So for example, all of the historic data for the RNA-seq profiling and all of the prospective data on the RNA-seq profiling, those are all going to be run on the same platform. And so we'll be able to amass that data over time and mine that data over time. And the trick is going to be getting as many different types of donors into that as you can, so that you might begin to see differences in genotyping those donors, for example. So maybe you begin to see differences in the PNPLA3 positive population versus not. But it's going to all start with building the database and populating that with the information. And so it'll be the first step in a long journey. But we do believe that donor homogeneity is out of the question, and that we will always be dealing with variability. And one of the things we want to be able to provide our customers is a more surgical way to select the donor-derived cells that will work in their application or address their specific needs. And that's kind of where we're trying to head.
Okay. That's all we have time for today's webcast. Please watch for our follow-up email with a link to view a recording of today's presentation on our website at samsarasciences.com. And on behalf of Dr. Presnell, myself, and the team here at Samsara, thank you for tuning in. Have a great rest of the day, everyone.